Well, I'm delighted to be here with Mr. Nick Inston from Birmingham in the United Kingdom, and also Dr. Kate Steiner from Stevenage. To make it globally important, we've got Dr. Dolmatch, Dr. Bart Dolmatch from Palo Alto in California. And we're here to discuss everything that's interesting and current when it comes to vascular access. And we've just had some breaking news that Ellipsis is being withdrawn. Nick, do you want to just fill in a little bit and get your colleagues to comment on this?
It came as a great shock, really. Like last week, around the world, we heard that Ellipsis, which Medtronic have been developing, really, endoAVF devices that we've developed data from, and really we thought was getting very established as part of practice. And I don't know if my colleagues have known any more information about it, but I'm a bit surprised that it's not carrying on longer.
So Nick's surprised. Kate, you obviously use endoAVF in your treatment armamentarium. Well, what are your thoughts on this?
Yeah, I mean, we've had an Ellipsis program for 2 years now at our institution. I think we've done about 30 patients. So, I mean, from my perspective, we were quite shocked by the news and disappointed from a patient's perspective. It's become it's kind of part of our algorithm now, part of our MDT working is we have a discussion, is a patient suitable for an endoAVF? And we have a number of patients who it's been their preference, as another number of patients where it's been what we think is the best treatment option for them. Admittedly, it's a smaller percentage of our total population having fistulas, but I think shocked, disappointed, and then wondering where we go now. Is it going to be to the next generation of devices? Do we change our personal use and change device? So a lot of questions and a lot of debate, and it's really fresh news. So we haven't had a time to get together as an MDT to discuss it.
Before I move to Bart, you're obviously very happy with how the device performed. Do you have any inkling as to why this product's been withdrawn?
Well, I mean, my inkling will be kind of speculation. I think it's the kind of product that when you adopt, it's not just the procedure, it's not just the product, it's the whole thing round it. It's having a team able to deliver a successful endoAVF program. And to do that involves a lot of education and a degree of training around cannulation in order to be able to do that. And I just wonder if that's something that we can do fairly easily at some institutions, but at others there may be barriers towards adopting a program and having it as part of your algorithm.
So that maybe some barriers to adoption, and maybe the reasons, therefore, it doesn't become standard of care. And we know Medtronic likes to invest in technologies that become standard of care. Bart,
And also,
sorry, Kate, what were you
I was just going to say the financial aspect of it as well.
Just tell me about that. How much does one of these cost?
So it's about
NHS?
£4,000 per catheter in the NHS.
4,000? That's quite high, isn't it?
Yeah, it is. It is.
Okay.
And I think that's one of the big problems we've had in the UK. It's not NICE approved. And setting up a business case for this is very difficult.
Yeah.
But other countries have done this. Rob Cheverdian in Germany has had a big program. Alex Malios in France has had a big program. So certainly this has been done elsewhere. I don't know what happened in the US. We thought that once this was taken on in the US, it was going to really take off as a big practice.
But that's interesting, isn't it? Because you've mentioned France and you've mentioned Germany. They're insurance-based systems where they have adopted and use this in larger volumes. And we're in a tax-funded, centralised system where NICE essentially hasn't said it provides cost-effectiveness. Bart, over to you. What is going on? Medtronic, we hold you responsible for every decision in Medtronic. What's going on with this big global US titan?
Well, it's a good question. I mean, I think Kate's final comment about the finances are certainly there. I think the adoption of percutaneous fistula creation has been relatively low, although I would say the Ellipsis has the lion's share of it globally. If you really look into some of the market analyses, something close to 85 to 90% of the percutaneous fistula market is the Ellipsis. And yet, I suspect for Medtronic, it just wasn't possible to show it as a profitable endeavor. Low adoption rates, certainly introduced kind of through the pandemic, partly because of that, but also creating programs that push this into the hands of interventional radiologists and interventional nephrologists. These are all kind of issues that slowed the adoption. And I think financially, if you're looking at it from Medtronic standpoint, you can't keep on running a program that may not be making enough money to support it year after year. I mean, that's not what rewards, senior vice presidents and CEOs.
Okay, so there's got to be a return on the investment, and there's been an investment in. Was it Jeff Hull who's the main person who developed this particular technology? Is that right?
He's listed as the co-inventor of this, but once Medtronic took it over, then I think it kind of went into other hands to develop it. And Jeff is
they need a return on that investment, don't they? They need a return on that investment. But what you say, that's very interesting, and I didn't know that, they hold an 85% share of the market. So this is a globally dominant player in a very small emerging technology. Does this mean the end of endoAVFs then, Bart?
No, well, first off, I would say that both devices, both the ellipsis as well as the wavelength, having Shaverdian has shown a very high rate of creating percutaneous fistulas. The issue is just the maturation, the point of getting them to be functional for hemodialysis. So we know the technology works very well to create a fistula. The downside is it just took a lot of surveillance and secondary procedures to really get most of these fistulas to be ready for cannulation for hemodialysis. So I think that, with I think advances perhaps in a second generation device that might reduce the number of procedures, it would be a much more palatable option to start a percutaneous fistula program.
But you're involved with the next generation device. Do you want to talk a little bit about that?
Well, yeah, sure. I am involved with the Venus 2 study, which is the Velocity. It's a small fenestrated covered stent that's used to create the fistula. Does not require any thermal or radio frequency energy. It's really a very simple procedure. The average length of time to do this is somewhere between 8 and 20 minutes or so. Simply puncturing like an ellipsis and deploying this fenestrated covered stent. The Venus 2 study is underway, more than half enrolled. It's a U.S. feasibility study, not the pivotal study that will ultimately follow. But I can't really share much of the results other than say that so far I think we're quite pleased with how things are going.
So there's early promise.
Look forward to concluding it.
So there's early promise. Too early to tell whether it'll be game changing. But what about price? Do you know what the price point will be? Has the company given you any indication? Will it be cheaper than ellipsis and wavelength, or more expensive? Or
I can't speak at all to that. No information. I will say that the reimbursement rates in the U.S. for ellipsis are quite good, robust through the coding system right now. So I think if they're not more expensive, there should be reimbursement for the device.
Okay, Kate, any thoughts based on what you've heard? I mean, between the two technologies that you have access to, how much of your usage was Medtronic versus the BD endoAVF?
We only had approval for the Medtronic, so our usage was 100% Medtronic.
Ellipsis. So this is a major setback then to see them with.
It is for us. Yeah, it is. I mean, we live in hope. Perhaps there's a company out there who might be interested in purchasing the technology. I mean, I don't know whether those conversations are happening in the background, Stephen. It's be a difficult position if a large global company like Medtronic have withdrawn it, and essentially it's kind of not working for them in terms of their strategy and perhaps financially.
Final thoughts, Nick, on this?
Well, it'd be really interesting to see what happens with wavelength, because this could, as you say, be the end of endoAVF, but equally it could be, a concentrated interest in a single device.
Yeah. Well, that's fascinating breaking news, and I'm sure we'll be discussing this more at Charing Cross at the end of April. But looking now at the future of how we use drug-coated balloons, particularly in vascular access, we started off getting very excited by paclitaxel, as we did in all aspects of the periphery, and now there's a new wave of Limus drugs. Do we want to get Bart perhaps to talk a little bit about what you see as the role of Limus drugs?
Sure. I mean, first I would say Limus is not my expertise, but I have read up on it recently and had some interesting discussions about it with people who know much more about it. Of course, the coronary stent market, has entirely flipped over to sirolimus type of drugs away from paclitaxel, recognizing that the mechanism of action is different. Limus drugs are static, whereas paclitaxel is cytotoxic and a bit more inflammatory. So, the results with the Limus drugs and the coronary arteries has been quite good. Whether this translates into some sort of application for AV access is still a bit unknown. There are different ways to approach this. But let's just talk a little bit about what we're treating. I mean, the goal here is to treat an AV access stenosis, with something that doesn't leave a mechanical scaffold behind, and yet prevents, neointimal hyperplasia. But we're not sure that that really happens anyway in all cases. I thought maybe Kate can share some of her information about what we're actually treating and when we expect to see neointimal hyperplastic restenosis.
Kate,
Yeah,
perfect
thanks,
intro.
Mark.
What are your
Yeah,
thoughts?
thank you, Bart. Thank you, Stephen. Yeah, I'm really pleased that this year at Charing Cross we will be sharing the results of our AV Sonopath trial, which is completed, and that was correlating the histology with the ultrasound findings, and looking essentially at that kind of radiology pathology correlation. So we know what we're treating, and we direct our treatment a little bit more towards it. One of the interesting things about the Limus is its anti-inflammatory effect, because what we have seen in some of the specimens, in particular those post-angioplasty, is not only hyperplasia, but an inflammatory reaction, which is interesting. I think one of the trials that's going to be pivotal in how we move forward with this is the PAVE-2 study, which we just started recruiting to, comparing plain balloon angioplasty with sirolimus and paclitaxel. And as part of that study, we're going to be gathering data on the ultrasound appearances, so ultrasounding in some of the centres immediately after, and at 3 months. I think that's going to give us a little bit more information about how lesions respond to anti-proliferative therapy. And of course, those where there's no intimal hyperplasia, do we actually still need that anti-proliferative therapy? What's the role? We don't quite know yet, but I think we're getting there.
That's fantastic.
I
That
think
you're going to see
that's
these data at Charing Cross. Bart.
Yeah, no, I think that's great, Kate. You say give a little bit more data. I think this gives a lot more data. Right now we just treat everything as luminal narrowing, and we presume
Yeah.
it's neointimal hyperplastic. But Kate and others have demonstrated that's not the case. There are certainly negative remodeled fibrotic stenoses. There are valves. There are hyperplastic issues. Sometimes inflammatory changes in the vessel wall. So I mean, it's really, we're 25 years behind the coronary endeavors to try to figure out what's going on. But I think it's finally time to figure out what we're treating, and in that regard, just decide whether, a drug-coated balloon makes any sense, for all lesions or just for certain lesions. So I think this is a great study. The PAVE-2 study is a great endeavor.
Well, just before I go to Nick, who's got some other technology that's in the Limus area, surely if we look at the coronaries, my memory was that sirolimus was the first drug-eluting stent, and then it went to paclitaxel, and then it's gone to other Limus drugs like everolimus. What basis do we think that paclitaxel is going to be superseded by Limus drugs? What's the sort of the presumption around that, other than what you've said about how it's what you said, Bart, about how it's got a different mode of action to sirolimus?
Well, the jury's still out about whether a sirolimus or a related Limus drug balloon is going to be effective or not. Again, a different drug, very hydrophobic, not inclined to stick around tissue very long. Will a balloon really impart enough drug to really have an effect on preventing neointimal hyperplastic restenosis? Yeah. We just don't know. So
we need these studies, like Kate, to get the answer. Nick, you've got a new technology.
It's on the same point of the pathophysiology of these devices, how these drugs work and the mechanisms. And I think it's a big challenge to treat stenoses, which we think are different. One of the sessions we're actually talking on in Charing Cross is, are stenoses all the same? And that's going to be different cases. But what we're looking at from a surgical point of view is maturation, so altering the cells before they form stenoses and actually getting in early. And there's a wrap device that we've got that we're going to do a podium first at Charing Cross. I can't give you the data now, so we're going to have to come to Charing Cross if you want to hear that.
And what's the study called?
And that's the Access-2 study.
It's the Access-2 study.
Vascular Therapies are running that from the US. But that's going to be really exciting because that's a randomised controlled clinical trial, blinded, very well constructed trial.
Wrap against what?
That's wrap against standard surgical creation.
Okay, so that's interesting. So randomised data, yeah. And you're presenting the results.
So I'll be presenting those results, and I don't know those results yet. So,
well, that's going to be very exciting. So 2 great bits of evidence that'll be unveiled at Charing Cross. Sounds a really exciting programme. One of the big debates at Charing Cross is whether drug-coated balloons should not be the standard of care. Sorry, it's a negative. Drug-coated balloons should not be the standard of care. Who's up against who, and what would you say is the answer? Should they or should they not be the standard of care?
So we've got a four-way debate on that. We've got it's not just one head-to-head. This is
it's a four-way.
Four people supporting different aspects of the argument. So I'll hand over to Bart, because he's probably got some ideas of whether or not they should be or should not be.
Yeah, no. First off, I would encourage anybody who's interested in this topic to come to Charing Cross. I think that the debate should be pretty robust. And, we go back to some of the concepts that we were just talking about, or drug-coated balloons. What's the agent? What's the mechanism? Is it effective? Do all lesions need to be treated? Which one should be treated appropriately? These are all up in the air right now, and again, the PAVE-2 study may answer a lot of this, but we really need to get more data on it. But overall, I'd say, it's taken, in my mind, a meta-analysis of all of the paclitaxel drug-coated balloon studies to try to figure out if there's a beneficial effect or not, and the meta-analysis shows some beneficial effect, but I think we're looking at a heterogeneous sort of lesions that are being treated. If you took out all of the negative remodeled fibrotic ones, you might see a much better effect, so there might be a specific role for drug-coated balloons, but I'm not sure across the board that they're really our first-line therapy at this point.
Interesting. So what are the four strands that we're looking at?
I'd have to look at the programme. Sorry, I've forgotten off the top of my head. Kate, can you remember?
Well, give me another strand. Give me any strand. Okay.
I know that I'm doing this with Narayan Karunanithy, who's, I think, the PI for the PAVE-2 study. Is that right, Kate?
Yes, he is.
I think one of the key features there is that the three studies on paclitaxel actually had very variable results and didn't necessarily support each other.
So there's no necessarily a class effect then.
And the PAVE study was in contradiction to two of the industry-led studies, which there's a lot of discussion about still. But as Bart says, that's been meta-analysed and still shows a signal. It's how we use them properly, I think, will be the key elements that come out of the debate.
Okay, so you'll be able to tease out the data and what it means and how you optimise.
I hope so.
That for the best management of patients. That sounds really fascinating, and it sounds like it's still very much an open field, and you do need the data, to get to grips with the data to get the right answers. Should we move on?
Yeah, I would say, let me just say that, initially we had small single center studies, that showed a good signal. And we said, but we need more data. Then we had robust, large, multi-center randomized controlled trials. And that gave us more data, but it was controversial as to whether there's really a benefit or not. As Nick mentioned, one study showed great benefit, another study showed no benefit. So I don't think we can advocate for more data. I think we just have to take a look and see what we're treating and decide where the benefit is for drug-coated balloons.
Got it. Well, I mean, perhaps I'll ask it in another way. If drug-coated balloons are not the standard of care, as this motion is intimating, what is the standard of care?
Based on
today, or is there no standard of care?
Yeah, is there? I don't think we've got the algorithm yet, Stephen, because it's not. I don't think it's like you treat this lesion. Every lesion's different. The location is different. Responds differently. So I think if once we get a standard, it's going to be a consensus around a kind of treatment algorithm.
Yeah.
That I think it's going to reflect a little bit more that Kadoki, right treatment, right patient, right time.
Okay.
Perhaps we're going to move to something a little bit more individualized. And of course, with the DCBs, there's another cost issue as well, isn't there? It'd be nice to see some really nice, robust health economic analyses around their use.
I mean, I think one of the interesting things is there's a new device called the Flex device, and I don't know if that's a third part of this discussion. That how is that going to affect either DCBs or plain balloons in terms of the delivery of the drug and the way that the lesion's disrupted, especially those fibrotic lesions? I don't know, Bart, if you've got any views on that.
Well, vessel prep is kind of a new concept for AV access, right? I mean, we've talked about it a little bit over the past couple of years, but, does vessel prep with something like Flex give a better outcome after Flex and balloon angioplasty? And again, we have some data that suggests, yes, that's the case. But, as to Kate's point, there are different sites of stenosis, different causes of stenosis. And I think until we better understand what we're treating, we're not going to have really a good algorithm to say what best to do next.
Just for my interest in the area of vascular access, dialysis access, what are the main forms of vessel prep? I mean, certainly if you look at peripheral arterial disease, there's a lot of interest in intravascular lithotripsy. There's a lot of interest in still, whether scoring balloons will come back. But can you give me an idea of what you consider to be the main options when it comes to vessel prep?
Yeah, so maybe people can help me here, but I would say plain angioplasty is vessel prep for drug-coated balloon.
Yeah.
Okay. Flex is vessel prep for plain balloon angioplasty. We don't know if Flex is vessel prep for drug-coated balloon or not at this point, but that's something that could be looked at. Beyond that, I think there's the options we have. We're not looking at laser ablations and other things right now. I think right now the goal is just to use either a plain balloon to prep the vessel or Flex.
But I think that's an important point in terms of why we're having the discussion against drug-coated balloons against plain angioplasty balloons, because to use a drug-coated balloon, you use a plain angioplasty balloon to start with. So they're kind of not comparable treatments to that extent. And it's just whether or not people will go in with a plain balloon angioplasty first line and then look and wait and see if they then use a drug-coated balloon. Yeah. Look at the response of that plain angioplasty and then use a drug-coated balloon, or actually just use a drug-coated balloon first line.
Interesting.
And I'd also mention that, these are all very highbrow discussions. These reach up to like what are we treating? Why are we doing it? What are the algorithms? But, when you come down to the practical level in the United States, which is, realistically, some people like to see patients back every 3 or 4 months. It just rings the cash register, and trying to do something that gives better outcomes is not necessarily in line with the economics of the situation. So there are some headwinds to try to use more expensive devices that may or may not be reimbursed.
Can I throw out something which may be completely wrong, but I thought, just based on a talk that I heard that Eric Sasemski gave about IVUS, because you mentioned how POBA is being used as vessel prep for DCBs, and he said that he uses IVUS because it often detects dissections that you otherwise wouldn't detect. Sure. And does that have relevance when it comes to dialysis access?
So I think IVUS is being used, and Kate and Bart may be using that, but it's expensive. But that might give us some more characterization of what the actual lesion that's being treated is. I don't know, Kate, I think you've done some work on it. Oh, Bart,
I think,
yeah, go ahead, Kate.
Yeah, I think one of the things about IVUS is, do you really need it when it's right under the skin? No.
Okay.
Where you're, treating a forearm, and I do a lot of my fistuloplasties under combination fluoro and ultrasound guidance or pure ultrasound. And you see a lot more with the ultrasound, I think. You do see those small areas of dissection, which you may not see on a DSA because you've got contrast, within the vessel obscuring some of the finer detail. I think IVUS is really, really useful for those more central lesions, which you can't see so well on ultrasound. So a deep cephalic arch and the central veins. Yeah. But for the peripheral, I'd kind of say, why would you? But you do see a lot with ultrasound. And I think you could see when you've got a really good angioplasty, size your balloons really well using ultrasound. And we've recently used a bit more in the vessel prep way for some of the calcified lesions. I've used some of the IVL, and I guess vessel prep, a lot of it is about get a really nice result. And we're learning, aren't we, a little bit from the peripheral vascular about how important vessel prep is before DCB use.
Very interesting. So again, there's no simple answer. It depends where the vessel you're treating and how deep it is. And the more superficial, it's less relevant. That's very interesting. Listen, I think we're going to have to switch topics again, and the next topic is around the new data on high flow fistulas. And over to you, Nick. Just if you want to introduce this.
This is a fascinating area that we particularly ignore. We're all concerned about getting a fistula running, making sure it's fit for dialysis. But very few people look at fistulas and look at the effect they have on the actual patient. And one of the best things I heard in a conference was a patient stood up and said, "You've created a fistula, you've created a new disease," and it's actually true. To have a fistula that's running at 2 litres, 2.5 litres, is the same as sprinting on the spot 24 hours a day for a patient. Wow. It has cardiovascular effects, but we don't really know. We can't get rid of the fistula, need the fistula. And one of the areas that a study's come out recently, a big study of about 16,000 patients in the US, of patients who've had transplants and then subsequently had their fistulas tied off, which is about 700 per those patients. And it's actually had no effect on mortality or morbidity. So we really don't know what to do with high flow fistulas. But they probably contribute to stenosis formation, they probably contribute to patient morbidity, and I think we need to monitor fistulas much more, particularly after transplantation, to see what to do with them. I don't know if either Bart or Kate particularly has a strategy with high flow fistulas, or we just tend to ignore them. It's in a lot of places, it's kind of supervised neglect. We measure them, we write down the high flow, and then we wait until something happens to the patient rather than actually intervening, slowing that treadmill down.
Well, before we go to them, what do you do in your practice?
What do I do? I want to set up a clinic now for post-transplant patients at 6 months and start looking at what we should do. I think there should be a trial on that.
Okay. So
Part of the Kadoki guidelines are that patients should have a life plan, a fistula life plan. When they have a transplant, they disappear into another system. No one sees them again. I had a patient come into clinic the other week who had two fistulas running at 3 litres each. That's triple cardiac output for what they should be doing. And the guy couldn't walk across the room because of this. And no one was doing anything about it. And he came in for a completely irrelevant thing, and I looked at his arms, and he had these 2 huge fistulas. So I think we need to look at them and start to develop some algorithm for what to do with these things, whether that's surveillance, whether that's banding, whether that's flow reduction, whether that's ligating fistulas and creating a new fistula.
So this is all starts off with more rigorous follow-up and then intervening in the right way.
I think so, yeah.
Okay. So Kate, Bart, any comments? Kate, what are your thoughts on high-flow fistula?
I think, Nick's outlined it really nicely. We get asked to scan a lot of the fistulas to look for high flow. One of the other problems that we see is, you can treat an outflow stenosis, and if the fistulas are high flow, even a small reduction in lumen means that that outflow stenosis becomes symptomatic. So addressing the high flow is really, really important. And I think, it's under recognized sometimes, even with patients who are on surveillance with fistulas.
Interesting. Bart.
High flow is, when you look at these dialysis patients before transplant, the biggest risk that they have, biggest mortality risk is cardiovascular. And it's sort of it just intuitive makes sense that a very high flow fistula would be disadvantageous, although it sounds like the data is not really showing that. As far as how you reduce flow, that's still a conundrum, in fact, yesterday, one of my partners banded a fistula and reduced the flow from 3 liters to 2.4 liters. I don't know. That's, how do we get it to the right point? How do we prevent banding or restriction of flow to prevent thrombosis? These are just still out there. We don't know how best to manage these high flow fistulas to get the optimal flow and prevent subsequent consequences.
Sure, but I think that's that's what one of the things for innovation, isn't it? We don't have a great solution, so that's a great point to start for what. Find out the problem and fix the problem. We've done it in lots of other areas in vascular surgery. And I think it really is a bigger problem than we think. And the high flow fistulas are easy to cannulate. Nurses like them. They don't cause problems until they cause problems, and the real problems. So I think we really do need to start opening the dialogue on this and start focusing more on it.
Just before we, I think that's a very interesting discussion then. So it looks like it's an open field and you need to start to build the research around how best to manage these patients with high flow fistulas. So just moving on to, before we move on to the Charing Cross, I was quite interested in what you see in the field as the most exciting things to keep an eye on. Perhaps I could ask each of you in turn, Nick, if with what we've been discussing up to now, what are the things that you're most keen to resolve over the next 12 months?
I think the data on the Access study is going to be really exciting because that will be the first pharmacological innovation that we will have seen. We've seen a number of ones that haven't worked. If we can find something that improves maturation, which is a huge problem, 30-35% of fistulas don't mature. We've got mechanical devices that may help, such as VasQ, but we've not got a pharmacological device.
Well, that's a real shift then.
There's not one drug that we can use to treat this, isn't it?
Well, are we on the cusp of a pharmacological management? That's very interesting. Well, Bart, before you go, any comments? What would you focus on?
First, the long range, I'd have to say I'm very intrigued by what's going on in xenotransplantation. Let's all admit that if you look at these hemodialysis patients, it's a miserable life, right? I mean, it's if there's something that could be done better, why not? So there's actually very early work that's being done now on gene editing for porcine transplant kidneys, and a couple of early failures, but certainly this is a topic that's not new. This has been around for decades, but I think with gene editing, I think we may see more rapid advance in the world of xenotransplantation. As far as hemodialysis access, Again, I think this entire story of what's going on with percutaneous fistula creation is really interesting, because we know we can make these fistulas with a high level of safety. It's just getting them to mature, and I think that the Venus 2 and then beyond that, the pivotal trial of the Velocity device may actually lead us into a second generation of percutaneous fistula creation. I'm going to have to go,
Yep.
Stephen.
Well, thank you very much indeed for your contribution. That's been much appreciated, and look forward to seeing you at the end of April in London at Charing Cross. All the very best.
Sounds
Bart.
great. Can't wait to be there. Thank you very much. I appreciate the time.
Cheers. And Kate, finally, just to finish off this point, what should we keep our eyes on?
I think we keep our eyes on that evolution of the treatment algorithm. Stenosis location, stenosis type, what's the best endovascular treatment, what's the best strategy for the patient, what gives us a good outcome for the patient, and also what's the best strategy in terms of kind of looking at the wider picture and the health economic analysis.
Great. Well, hopefully we'll see big advances in an evidence-based algorithm over the next 12 months. Well, listen, we're obviously very excited by the Charing Cross. It's become probably the largest vascular access and dialysis access meeting in Europe under your stewardship, both you, Nick, and Kate. What are the highlights going to be? What should, for those who are coming, and we had around 3,000 people last year at Excel, and we hope to have a similar number again. What should we focus on? Perhaps start with you, Kate, and if colleagues could help Bart to leave. I think he's looking to leave. That'd be great.
I think for me, we've got so many podium firsts this year. Weren't we kind of turning them away almost?
Yeah.
Lots of great late breaking data, really good high quality data, and the great debate. I think the DCB debate for me is going to be really lively, especially after our last debate that we did. Well, I think we're quite brave at Charing Cross now, aren't we? Addressing some of the real problems that we see in practice. And last year, I think we were brave when we did the debate about the endoAVFs and was this a failed experiment? And I think it's really good to have these controversial debates that are on people's mind and asking the questions, because it makes it so much more relevant.
Well, what's very interesting, because that topic, is it a failed experiment, was once used about EVAR, and EVAR carries on being relatively successful 40 years after the first stent graft was implanted by Volodos, and yet, it does look prescient to have had that debate a year ago with the withdrawal of ellipsis. So fascinating.
Yeah, absolutely.
Yeah. Well, I mean, what you say about Charing Cross is, certainly when my father started it as a much younger man than I am today, it was about education and innovation, but also about the evidence. And I'm so pleased that there's so much new data. But Nick, what about from your standpoint as a...
So I think the biggest challenge at Charing Cross is for me and Kate to keep time, because there's so much discussion goes on.
Well, how do you do that?
Well, we have to... we keep
do we need to bring back the bell?
Yeah, we'll keep the sessions short, and we'll try with it's actually such a broad subject. I've had people say to me, how do you keep a full day going with about vascular access? Isn't it just one 10-minute talk? But there's such a vast area, and renal patients are such a challenging group that most vascular surgeons and interventionists will at some stage interact with a renal patient. And I think what we're trying to do at Charing Cross is not only look at those people who have big practices in this area, but also find the people who are maybe in district general hospitals, smaller hospitals, who have small groups of renal patients, but also for them is a huge challenge. So we start off with maintenance, creation, the indications, the timing of when we create fistulas, which is a big topic. We've got Charmaine Locke coming across from Canada, who wrote the Kadoki guidelines, who's going to go through that. That's a huge issue, because we create a lot of fistulas that we never get used. And we're actually doing patients probably a disservice by creating fistulas too early. But we also have a lot of fistulas that don't mature, and people start on central venous catheters, which we know are detrimental in so many ways, quality of life and quantity of life. We're then going to move on for some of the technologies, and we have a very good relationship with industry on this, and I think vice versa. Industry work closely on patient benefit, and a lot of the endoAVF work we've done has been to try and see if it's beneficial for patients, if patients prefer this, if it's less hassle for patients and things like that. So that comes back to the disappointment why, if endoAVF is pulling away from the market, that a lot of patients actually really like that. And
I just wanted to tease that out, because I didn't at the time. You've now mentioned that a lot of patients really preferred endoAVF over other options. Kate mentioned that it was, essentially the device of choice as an endoAVF ellipsis, and many patients preferred it. Why is that?
So one of the reasons is that the fistula actually flows through different veins. So it comes from a deep vein system into superficial veins, and actually flows through two rather than one single vein. We think that higher flow, back to the higher flow argument, is detrimental to endothelium, especially when it goes around corners like at the central venous, the cephalic arch, or at the swing segment. And we don't see that in the endoAVFs. So you're not seeing the same number of stenoses creating, you're not seeing aneurysms, you're not seeing high flow fistulas. So
you're saying the problems essentially at the end.
I don't think we've seen many aneurysms in these. We've not seen cosmetic issues. There's a study done, the AVAC study, looking at cosmetic issues, and patients preferred endoAVFs over standard fistulas. So there's all of those benefits there. And whilst there is a reimbursement issue, it's a shame that patients miss out on that because of the politics of reimbursement.
So here's a technology that's working, that patients really appreciate, and it seems to be foundering on the price point, essentially.
I think so, yeah.
Okay. Kate, anything to add on that patient preference for endoAVFs?
Yeah, so we had a number of patients that started in the early days where patients are really well informed now. They'll look online and asked for an endoAVF, predominantly for cosmetic reasons. And then once we had a few patients in the unit with the endoAVFs, you'd have other patients say, "Oh, your fistula looks different. You know, have a look." And I'd like one of them. So friends of a friend self-refer themselves for an endoAVF.
Wow. Interesting, that word of mouth. Fantastic.
Yeah, absolutely.
I mean, the other thing I'm really excited, we've discussed it a little already, is the case studies. We often stand up and just present, and Bart's an excellent advocate of this, stand up and put a venogram up and say, what would you do? And we have, the aisles are full at the microphones, people arguing about what they would do in real practice. And these are real world cases. We don't just stand up and present obscure stuff. And often you can get into a discussion for 20, 30, 40 minutes about something that you think is a really simple issue. And it depends on where people are in the world. And of course, we've got a global audience at Charing Cross. If you've got the devices that are available in some countries and you haven't in others, you have to look at different ways of doing things and innovate.
Yeah, so you've got, as you say, lots of podium firsts, you've got the debates, you've got case-based discussion and presentation of very exciting, interesting cases, but you also have got a workshop, and the Vascular Access workshop is very popular. Do you want to talk a little bit about what we expect to see this year?
So it's not just one workshop. We've done it in various formats this year. What we're going to do, have booked on sessions, and I think we've got 5 different workshops at the moment, looking at endoAVF and surgical AVF. We're doing a trainee workshop. We've got a lot of feedback saying trainees wanted to actually sit down and learn how to do a fistula from people who can do them on jigs. We're doing some work on devices for thrombectomy. And I think, are you involved in that one, Kate? Or I certainly think Bart's involved in that one.
I'm not sure. I think Bart is. I'm certainly involved in the one. We've got some patients coming again for ultrasound. So we've got the ultrasound workshop.
And that's hands-on with patients, mapping and actually doing ultrasounds of fistulas, which are a very difficult lesion because, again, they're not normal. These are anatomically very, very abnormal looking things. So if you're a sonographer who doesn't look at these, it's very difficult to do things like measure flow in the fistula, and there's tricks to get around that. So fantastic.
So you're getting to learn and practice lots of tips and tricks.
And ask the experts. It's a real opportunity, I think, with the workshops. Often people will have an opportunity now to speak to one of the leading experts in a particular area of AV access, or almost a one-to-one.
But again, we also get industry involved in these, and they bring state-of-the-art simulators. So you've got, benchtop simulators, so people can practice over and over in there. And rather than being at a stand where it might be a bit more pressured, they've actually got the time to be taught and tutored in those sessions.
Fantastic. So the just want to remind people when the days are. So when do we expect the vascular access and the renal interventions day to be? What day is that? Is that on?
So the main workshop, the main masterclass is on the Monday. On the Wednesday.
Yeah.
I keep getting confused.
On the Wednesday because it's Wednesday, Thursday, Friday.
Jump one day forward.
Yeah.
On the Wednesday, and the workshops are on the Thursday.
Okay, so something going on for the first 2 days of the symposium.
That's right. Yeah.
Brilliant. Any parting thoughts? I mean, it's been a fantastic, action-packed discussion. I really appreciate your time and also Bart's time. Kate, any closing remarks about CX and or anything at all?
I think it's really about the ethos of CX, and I think last year we saw really great contribution from the audience. We really want to get people along so they can express their views and join in. So it's very sort of inclusive. and environment at CX, and I think that generates a lot of lively debate.
That's fantastic. And Nick,
every year
finally.
at CX, we've been doing it for the last 10 years, and I go away every year with, I've learned new things, I've met new people, I've heard different ways of doing things, and I go away with new ideas that would translate back into patient care. So that's what I think everyone can take away from CX.
Well, I think that emphasis on new, and my father coined the phrase when we were 40 years in, that it was 40 years of looking forward and evolving and not looking backwards. And I think that's certainly what you brought to the Vascular Access Programme. And I think the remark that you made, Kate, that was certainly my father's belief, that you bring together a world-class faculty, but above all, it's that engagement with an expert audience on how to optimally manage things in the interests of the patient. So looking forward to seeing you both at the end of April, and also most of the world. We've got over 80 countries that typically attend Charing Cross. They're in person and many online as well. So thank you very much indeed for your time. It's been wonderful having this discussion, and I'm licking my lips. Next Wednesday on the 23rd, and the workshop on the 24th. Make sure that you're there.